This fish hasn't had a tongue in over a year. But it's still feeding, still swallowing, still pressing prey against the roof of its mouth the way every fish on Earth does. Because something else is doing the job. What you just saw is real. It has a name, Cymothoa exigua, the tongue-eating louse. It is the only known animal in the world that doesn't just kill its host's organ, it replaces it. It enters the fish through the gills as a centimeter-long swimming animal. It crawls forward through the gill arches until it reaches the mouth. It locks itself onto the base of the tongue, and then, over a series of weeks, it does something we still don't fully understand how a brainless crustacean can do at all. It kills the tongue, and it takes its place. By the end of this video, you will understand how a creature with no central nervous system performs the most precise act of biological theft ever documented on Earth. Let's start with how it gets inside. In the warm waters of the Eastern Pacific, from the Gulf of California down through the coast of Central America, there is an animal you would not look at twice. A small, pale, segmented crustacean, bug-eyed, about the length of your thumbnail. It belongs to a family of isopods called Cymothoidae, a group of roughly 280 species. Most of them are unremarkable. They cling to fish gills, they sip blood, they live quietly. But one of them does something none of the others do. Cymothoa exigua does not stay on the outside. When it's young, a free-swimming larval form called a manca, it drifts in open water, tiny, almost weightless, looking for one specific thing: a host. The manca is barely visible to the naked eye, about 3 millimeters long, almost transparent in seawater. It cannot see well. It cannot navigate by sight at all. What it has is chemistry. Along the surface of its body are clusters of chemoreceptors, tiny sensory hairs tuned to specific dissolved compounds. They respond to the chemical signature of fish mucus, to the breathwater of certain species, to the trace amino acids in fish urine. The manca doesn't search for any fish. It searches for one kind, the right kind. Almost always is a snapper from the genus Lutejanus. Other fish swim past unnoticed. But when the chemical gradient sharpens, when the right combination of compounds reaches the right receptors, the manca turns and follows. It has weeks to find one, maybe 4. After that, its energy runs out. Most mancas die in open water, drifting unfed and unmatched. The ocean is enormous. The signal is faint. The window is small. But the ones that find a host, and not just any host, the right host, start the next phase. When the larva finally bumps against fish tissue, a reflex fires in its claws. It grips. It does not let go. It walks inside. The space behind a fish's gill cover is a labyrinth. Filaments, arches, bony supports. To anything but the fish itself, this is a maze of soft tissue and sharp edges. Most parasites would die in there. But Cymothoa exigua has 7 pairs of legs. Each one ends in a curved claw designed to grip. It doesn't fight the current, it walks against it, pulling itself forward through the gill arches, one filament at a time. To understand why this is so strange, look at what other Cymothoidae do. Analocra species attach to the outside of a fish, on its side, behind its eye, on its head. They look like an oversized tick gripping a hide. They feed by piercing skin. They never go deeper. They stay where the water flows over them, breathe the same water their host breathes, drop off when satisfied or when it's time to molt. Neurocilla, almost the same story. Different fish, different anatomy, same logic. External attachment, surface feeding. A parasitic lifestyle that doesn't ask much of the parasite. Both of these animals evolved from the same ancestors as Cymothoa exigua. Both belong to the same family. Both had every opportunity to keep going further inward. They didn't. Only one species in this family pushes past the gills and into the mouth. Only one anchors to a hard internal structure. Only one replaces the host's tongue with itself. And here is where the journey of this one species gets stranger. Because while one Cymothoa is making the trip inward, others may be doing the same. 2, sometimes 3, occasionally 4, all arriving on the same fish, all moving through the same gill chamber. And what happens next decides everything about how the rest of this story unfolds. Cymothoa exigua does not have its sex set at birth. It is determined by who arrives first. The first individual to settle on the fish, usually the largest, the one to arrive earliest, begins to transition. Its body broadens. Its anatomy reorganizes. Its gonads convert. It becomes female. Every parasite that arrives after her stays male. They cling to the gills. They wait. And they wait close, because she only moves to one place. The base of the tongue. This is sex determined not by genetics, not by hormones inside an egg, by geography, by arrival. The first parasite to reach the host becomes the architect of the entire takeover. Every other parasite on that fish becomes part of her support staff. And now, she begins the surgery. You might assume the next step is the kill, that she devours the tongue, drains it, strips it down. But what Cymothoa exigua actually does is slower, quieter, and it takes weeks. To understand what happens next, you have to understand the tongue itself. A fish doesn't have a soft, muscular tongue the way you do. What sits in the floor of its mouth is a structure called the basihyle, a bony platform covered in a thin layer of soft tissue. It doesn't taste, it doesn't speak. It does one job. It presses food against the roof of the mouth so the fish can grip it before swallowing. That is the entire purpose of the organ. And the soft tissue covering that bone, that is what Cymothoa exigua targets. She doesn't bite into it. She doesn't poison it. She doesn't tear it. She holds it. Her terminal claws, those 7 pairs of hooked periopods, clamp around the tongue right where it meets the bone. They press into the tissue, and they don't let go. What they press into matters, because the tongue, like every living organ, runs on blood. A small artery branches off the ventral aorta, the main vessel beneath the gill arches, and runs up into the soft tissue of the tongue. That artery is what keeps the tongue alive. Cymothoa exigua's claws sit directly on top of that artery. What happens next is something every surgeon in the world has a name for: ischemia, the slow death of tissue that has been cut off from its blood supply. Ischemia is one of the oldest weapons in biology, not because it is dramatic, it isn't, but because it works in a way the host cannot fight. Toxins can be normalized, wounds can be sealed, parasites that bite can be inflamed against by the immune system, swarmed by white cells, walled off in scar tissue. The host has 1,000 defenses against active attack. But ischemia is the absence of attack. It is the absence of blood. There is nothing for the immune system to fight, because nothing is being added, only taken away. White cells cannot reach a tissue that has no circulation. Inflammation cannot reach a tissue with no flow. The starved tissue dies without ever knowing it was being killed. Surgeons exploit the same principle. To shrink a tumor, doctors sometimes block its blood supply on purpose, a procedure called embolization. To control a wound, they tie off the artery feeding it, a tourniquet. The body's most efficient way to remove a part of itself is to stop sending it blood. Then everything else takes care of itself. Cymothoa exigua discovered this strategy hundreds of millions of years before any human surgeon did. At first, nothing visible changes. The fish keeps eating. It strikes prey. It presses food against the palate the way it always has. The tongue still works. The parasite is just there, attached, riding along. But under the surface, the tongue is dying, cell by cell, layer by layer. The tissue, starved of blood, begins to wither, to shrink. Its color drains, its fibers thin. Over the course of weeks, sometimes longer, the entire soft tongue atrophies away from the bone beneath it. And the fish, during all of this, shows almost no sign that anything is wrong. Fish have nerves. They have nociceptors, pain-sensitive receptors, distributed across their bodies. They can feel injury. They react to it. But the base of the tongue is not a high-pain region. There is no behavioral imperative in a fish's evolution to monitor that exact patch of tissue for slow decay. A predator's bite, yes. A hook in the lip, yes. A slow loss of blood flow in an internal structure behind hidden anatomy, no. This is the deepest part of Cymothoa exigua's strategy, and it isn't strategy at all. It is geometry. The parasite happens to be attacking a part of the host that the host evolved to ignore. Slow parasitism is invisible by design. It is invisible because there is no design, only an ancient, blind match between an attacker and a blind spot. And then, one day, what's left of the tongue simply falls away. The basihyal, the bony platform, is still there. Cymothoa exigua is still there, but the soft tongue is gone. And here is the part that makes biologists uncomfortable. The fish does not seem to mind. It keeps feeding. It keeps swallowing. It keeps doing the only thing the tongue was ever for, pressing food upward against the roof of its mouth. Except now it is not pressing with its tongue. It is pressing with the parasite. Cymothoa exigua is the only known animal that doesn't just kill an organ, it takes its job. Look at the parasite's body. Smooth, curved, pale. Roughly the shape and size of the organ it just destroyed. This is not a coincidence. Over millions of years of evolution, the body plan of Cymothoa exigua has converged on something that fits the host's mouth almost perfectly. It anchors itself to the bony basihyal stump, the same place the original tongue grew from. Its 7 pairs of claws lock around the bone. Its body lies across the floor of the mouth. Its dorsal surface, the smooth, broad upper back, faces the palate above. And when the fish strikes prey, the parasite is what touches the prey first. The fish is feeding through the parasite, and the parasite is feeding from the fish. Most of each meal goes down the throat the way it always did, but a small fraction never makes it past the mouth. Particles wedge into the parasite's joints. Mucus collects against its body, and underneath, where her claws still grip the basihyal, Cymothoa exigua continues to sip blood from the tissue around the bone. Blood for her,
scraps for the males still waiting at the gills. She lays eggs inside the fish's mouth, fertilized by the males clinging to the gills only inches away. The juveniles drift out of the host's gill slits into open water looking for the next fish. The host, meanwhile, keeps swimming, keeps eating, keeps living.
For years. And for decades, this is where the textbook stopped. The line was always, the only known animal that functionally replaces an organ of another animal, and the host fish lives a normal life. That was Brusca and Gilligan's note in 1983, the paper that first described what Cymothoa exigua does. They were morphologists. They were describing what the parasite looks like and where it attaches. They were not measuring whether the fish was, in any meaningful way, fine. But the line stuck, decade after decade. Every popular article, every nature documentary, every wildlife photo caption. The fish lives a normal life with its parasite tongue. As if biology had found a strange, dark accommodation that worked for both sides. It hadn't. Starting in the 2010s, fish biologists began doing what nobody had thought to do for 40 years. They started measuring. They measured condition factor, a simple ratio of a fish's weight to its length. A well-fed, healthy fish has a high condition factor. A struggling one has a low one. They measured gonadosomatic index, the proportion of body weight devoted to reproductive organs. The more energy a fish has to spare, the bigger and more developed its gonads. The less energy, the smaller. They did this not just for Cymothoa exigua. Long-term data specifically for exigua is still thin, but across the broader Cymothoidae family. Wellicke and colleagues, other groups, sample after sample. The fish were not fine. Their condition factors were lower. Their gonads were smaller. Their reproductive output was reduced. On average, they grew more slowly and didn't live as long. The thing in the mouth was costing them, quietly, continuously, for the entire duration of their lives. So why did the science miss this for 40 years? 3 reasons. Cymothoa exigua doesn't kill quickly. There are no dramatic deaths to investigate. The cost is subtle. You need large, careful samples to see it. And the wow factor of organ replacement was so loud that nobody bothered to ask the quieter question. The headline was so strange that the truth got buried under it. We were wrong about the most basic part of this story. The fish doesn't live with the parasite. The fish survives it and pays for every meal it ever swallows again. But there is one question, even now, that nobody can fully answer. Cymothoa exigua has no brain. No central nervous system worth the name. So how, exactly, does it know how to do any of this? Think about what Cymothoa exigua actually does. It enters a specific kind of fish. It travels a specific path through that fish's anatomy. It positions itself at the exact base of one specific organ. It applies pressure on the exact right artery. It waits the exact right number of weeks. It anchors at the exact right point on the bone. And then it performs, passively by being there, the exact function of the organ it destroyed. Every step has to be right, and every step is performed by an animal with no brain. Cymothoa exigua has nerve ganglia, small bundles of neurons scattered along its body. It has sensory hairs. It has chemoreceptors. It can feel direction. It can sense chemicals in the water. It can move, but it cannot plan. It cannot map. It cannot decide the way a vertebrate decides. And yet it performs the most precise act of biological theft we have ever documented. So how does it do this? The answer, as far as we currently understand it, is that Cymothoa exigua does not know anything. It does not plan the surgery. It does not aim for the tongue. It does not measure its position. What it has is a sequence of behaviors, each one triggered by a specific chemical or tactile cue. Each step looks surgical because under those exact conditions, it is the only step the parasite is capable of taking. The free-swimming larva is attracted to certain fish chemicals dissolved in the water. It moves toward them. When it bumps against fish tissue, a gripping reflex fires in its periopods. It grips. The current behind it draws it deeper into the gill chamber. Once inside, its sensors detect surfaces, and it moves along them, always pushing forward, always away from open water. It exits into the mouth because that is the only place forward leads. When it senses the basihyal, a hard, unmoving surface beneath softer tissue, its program switches. The claws clamp. They lock. They hold pressure for as long as the parasite lives. The atrophy is not done by the parasite. It is done by the fish's own circulation. The parasite simply holds. And the becoming the tongue part? That is not deliberate either. The parasite anchors where the tongue used to be because the tongue's base is the only hard surface in the right size range. Its body sits across the floor of the mouth because that is the path of least resistance. When the fish closes its mouth around prey, the prey presses into whatever is there. The parasite happens to be the right shape, the right size, the right position to do what a tongue would do. It is not surgery. It is a sequence of behaviors so precisely tuned by evolution that the outcome looks like surgery. To understand how this happened, you have to understand the deep timescale isopods come from. The order Isopoda is roughly 300 million years old, older than dinosaurs, older than the first mammals. Most isopods are bottom feeders or scavengers, pillbugs on land, woodlice in soil, deep-sea giants on the ocean floor. None of these animals look like predators. None of them look like surgeons. The family Cymothoidae, the family Exigua belongs to, split off from these mundane ancestors much more recently. Roughly 150 million years ago, somewhere in the warming Mesozoic oceans, certain isopod lineages began attaching to fish instead of scavenging. The earliest forms were external, crude, clinging to gills the way Analocra still does today. But evolution did not stop there. Over tens of millions of years, branches of the family pushed deeper. The ones who held on a little firmer survived. The ones whose claws sat a little closer to a blood vessel, they survived. The ones whose bodies shaped a little more like the host's anatomy, they survived. The ones whose larvae were a little better at finding the right species, they survived. What we are looking at when we look at Cymothoa exigua is the long tail of that pressure. 300 million years of small mechanical refinements, generations of isopods that happened to attach a little closer to the mouth, a little firmer at the base, a little more shaped like a tongue. The ones whose claws gripped the artery survived to reproduce. The ones whose bodies fit the mouth survived to reproduce. The ones who didn't disappeared, leaving no trace. What looks like a surgeon is not a surgeon. It is a sequence of mechanical reflexes that, after enough time, became indistinguishable from a procedure. And this is why Cymothoa exigua is so unsettling. Because what it shows us is that surgery, the most deliberate, intelligent act we associate with our own species, can be reproduced by something with no mind at all. Given enough time, given enough generations, given a small enough pressure to refine each step. Evolution does not need to think to be precise. Cymothoa exigua is not the only animal that proves this. It is just the one that does it the most visibly. Somewhere in the Pacific, right now, a fish is feeding. A small crustacean inside its mouth is feeling food pass over its back. And neither of them knows what is actually happening. Only one of them ever will. If you're the kind of person who likes peeking behind the curtain of our eerie, beautiful world, the kind who wants to know what the textbooks left out, then this channel exists for you. The deep dives keep coming. The stranger ones haven't even started yet. Subscribe so the next one finds you. There is a deep catalog of these creatures ahead. Parasites that hijack brains instead of organs. Viruses that turn animals into killers. Fungi that perform their own kind of takeover. No surgery required. In the next video, we move from a parasite that becomes a tongue to a virus that taught the world what a zombie looks like, long before any movie did. It has been with humans for thousands of years. It has done all of it without ever having a face. You will want to see this one.